Hi, I'm Hannah. I'm from John Jay College of Criminal Justice in New York City. This summer, as part of the University of Florida's Summer Undergraduate Research at Florida program, I was able to study how insects respond to extreme temperatures on a behavioral and molecular level in Dr. Daniel Hahn's lab in the Department of Entomology and Nematology. Climate change poses a major threat to many organisms, from animals to plants. In addition to generally warmer temperatures, another dimension of climate change is an increase in variability in temperatures on the seasonal shoulders in fall and spring. This increased variability will include rapid temperature shifts from warm to cold and vice versa, as generally warmer conditions are affected by an increased frequency of cold fronts. The stress of rapid temperature shifts will be particularly felt by insects and other cold-blooded animals that will need to cope with temperature swings. If certain insects are unable to cope with these changes, population growth may decline, leading to environmental consequences such as a decrease in ecosystem services provided by beneficial insects like pollinators or decomposers. By studying how insects respond to temperature shifts, we can build our toolbox to predict how ecosystems may be affected when faced with climate change, and work to mitigate negative effects. Populations can be resilient to climate change if they can acclimate to new temperature regimes, or they can adapt to these new regimes evolutionarily. Whether populations can undergo evolutionary adaptation in the face of climate change depends on the presence of genetic variation for climate-relevant traits in the population. To understand whether populations have sufficient genetic variation to facilitate adaptation to rapid warming, we measured how different genotypes of fruit flies coped with either gradual or rapid increases in temperature. The fruit flies we tested were bred to represent worldwide genetic variation. We used 13 different genetic lines of fruit flies that have known differences in their genotypes. By determining their responses to temperature increases, we could relate those differences in thermotolerance to their genotypes and assess genetic variation. Flies with genotypes lacking genes that protect proteins from unfolding and becoming non-functional wouldn't also contain genes that are susceptible to protein unfolding. This suggests that flies capable of acclimating to one type of temperature increase may be vulnerable when exposed to a different rate of warming. Another result suggested male flies were more resilient to temperature increases than female flies. Because females are critical for reproduction, their susceptibility to high temperatures suggests that population growth may slow down or decline in the face of climate change. Further goals of this research aim to determine the extent to which more heat-tolerant genotypes have reduced protein unfolding and to characterize the underlying genetic changes that lead to greater protein stability. By understanding which fruit fly genotypes better acclimate and adapt to the anticipated increases in temperature, we may be able to prevent potential population declines in other insects as well. Thank you so much to the SURF program for funding me this summer, and thank you to the Hahn Lab for welcoming me as a SURF student. Thank you, Dr. Hahn, for being an amazing mentor, and thank you, Andrew, for being a great mentor and friend.